Okay, hello. So this is the last chapter of the course, and it's chapter 12, and this is all about metabolism and using food as fuel. And we're going to be talking about acetyl coenzyme A. We're going to be talking about ADP, ATP, again, these nucleotides that we just learned about in the preceding chapters. And we're going to be talking about digestion of proteins, carbohydrates, and fats. We are going to be talking about the citric acid cycle as a necessary part of that, as well as, well, what happens whenever the diets are a little bit heavier in fats and proteins. So that's going to be rounding out the rest of the chapter. So let's just get into it, and we're just going to go. So here's the metabolic overview, right? So you either have the breakdown of matter or the creation of matter, right? The decomposition or the synthesis. And in this particular case, we're going to consider the decomposition to be catabolism and the synthesis to be anabolism, all right? So this is large biological molecules versus the small building blocks, just like how nucleotides were formed from the combination of like pentose cyclic sugar plus the nitrogenous base plus the phosphate. Again, to make the nucleotide or nucleoside if you didn't have phosphate. And then, of course, amino acids linking together to make the proteins ultimately. All that is basically what this chapter is talking about. And so the necessary nucleotide that is involved in all aspects of this is going to be ATP, adenosine triphosphate, okay? And, whenever we first start to eat, obviously, this is from a macroscopic scale, and this figure that I have on the right here kind of shows that whether it's proteins, polysaccharides, or lipids, it doesn't matter as long as those nutrients can get into the cell, then they can further be processed by the mitochondria. And this kind of flow chart shows all of the different action. And we're going to be talking a bit about elements of this flow chart, including pyruvate, acetyl coenzyme A, the citric acid cycle, as I just mentioned, and also these coenzymes like the NADH, the FADH2, things like this. So this is going to be very common throughout this chapter. So if you see that we're generating some ATP and ADP and FAD and whatnot, you're in the right place. So this is what we're talking about. Notice that with the breakdown of large biological molecules, what I mean by that, the foods we eat, like proteins, fats, carbohydrates, the whole bit, whenever we catabolize them, electron transfer will necessarily ensue. And, in doing so, our bodies then use that breakdown of material right into the small building blocks for our own purposes to create new material. And that's really important, obviously, for our health and whatnot and for our survival. But one of the interesting parts about this is that, and it's usually not discussed too much, is that there is going to be transfer of electrons and all of that. And that is where the NADH, FADH2 really start to impart a huge role in that, because they help transfer those electrons. Those electrons are always going to be shuttling around the different aspects of the cell where the nutrients are going to be delivered. So it's not all in just in one core place. It's not like everything's digesting, for instance, in the stomach, the large intestine, or even the small intestine. I mean, this is going to go throughout our bodies, and so things are going to continue to be created as needed. And at that time, of course, you're going to have electrons being transferred. And so a large part of this whole process is going to be balancing the heat that is naturally produced as well as the electron transfer. So obviously we can't have a situation where our bodies become unstable and we get too hot and we explode or we become superconductive, right? Like a piece of metal. It's just not going to happen. So there's going to be a nice balance here. And what we're going to be talking about here is really an overview of that, and we won't get into too much detail with that fine structure in terms of where the electrons are going, but we will be discussing it. So I think that that's important to know. So these systems, and why we even have all these cycles and stuff, it's a great way for us to efficiently handle heat, and it's also a great way of handling electron transfer. Okay, so digestive system, cell, and mitochondria, and from your book, there's a table point 1 that talks about the different properties. Again, we're going to be talking about hydrolysis and condensation. You guys are going to become very familiar with that again. I know that goes back to chapter 5 material, but it never goes away, because if we're going to deal with that electron transfer, of course, we're going to be dealing with redox situations then. So here's a schematic here, a cartoon, if you will, of animal cells, and I'm sure this is a very familiar kind of structure for you guys. You have the cytosol, which is just a liquid part of the entire cytoplasm, which encompasses everything. Obviously, you have the nucleus, you have the mitochondria, right? That's the energy production. So that's going to be key in generating ATP, you know, making sure that also at the same time that things are heating up too fast whenever that is generated. You have all this endoplasmic reticulum structures, whether they are smooth or rough, right? And we give a little bit of discussion down here where it says that, you know, this process of proteins for secretion synthesizes phospholipids, right? Synthesizes fats and steroids. All of this stuff is so important. You have Golgi complex, the lysosome. You have all these other things. The ribosome, we just learned about the ribosome in more detail last chapter. I mean, that's where after some of these, you know, the mRNA is then duplicated to create the proteins. That's almost like the, one could consider that to be like the protein factory. It's comprised of both protein and RNA subunits that help to do that. And I'm trying to think if there's anything else. I mean, that's it, other than the cell membrane, which is going to have that phospholipid layer, right? So bilayer. So I think that's an important overview, and all this stuff is pretty important, and it's pretty interesting that the mitochondria, again, I'm going to go back to that bacterial part where the prokaryotic cell, you know, of bacteria, like for instance E. coli, has a lot of similarity with respect to the mitochondria. And because the mitochondria is so complex and it really dictates everything going on, it's, you know, I mean, and that's what we're going to be talking about today. You know, it's a really interesting comparison to be made for sure. All right, so we had already introduced ATP and ADP previously. Usually we introduce them as like coenzymes in certain types of reactions. And I know we kind of talked about that last chapter, although we were mostly focused on monophosphates with the nucleotides or nucleosides, but here we're going to be talking about the diphosphates or the triphosphates. Okay, so these are also nucleotides. It doesn't have to just be a monophosphate. It can be a diphosphate, it can be a triphosphate. And in this particular case, we have here drawn on the left the adenosine triphosphate and adenosine diphosphate. And so what you have here is the nitrogen space. You have the sugars, and then you have phosphate. Actually, I just realized this should be adenosine. Yeah, I mean, this is good. What's going to happen here is that you're going to have condensation and hydrolysis playing a major role, and you have a high energy form, meaning that one's going to have a lot more phosphate than the other form. I think that's the easiest way to look at this. And then the ADP is going to be considered the low energy form. You know, in the processing of ATP, whenever that phosphate is going to free up, well, guess what? That's going to produce some energy. So I think that is pretty important. That heat energy is what the mitochondria is especially really good at dissipating. I mean, it takes it and then it can use it immediately for something else, as well as the, you know, the rest of the cellular components in our body, because we can then, you know, further you know, construct things, whether it's anabolic behavior, whether it's, you know, helping to shuttle electrons from one part along the cell membrane to another part or to another type of organelle. All this stuff is very interesting and very helpful. So you have the adenosine base structure. As you know, you have the fructose, well, it's the pentose, cyclic pentose here, right? And it looks like the fructose molecule, of course, and the nitrogen base along with those phosphates. Now, the key here, and something that is going to be helpful for you guys to know, is that whenever ATP is basically hydrolyzing the ADP, it's undergoing reduction, right? And if it's going the other way, ADP will then oxidize the ATP. So with the larger molecule ATP, that will signify that you are getting a larger entity, oxidation present. Whereas, you know, what form is that oxidation? Well, you're getting a whole nother phosphate structure, which has negative charges, and it also has an abundance of oxygen, right? And I think that is one of the key parts here. With respect to the reduction, there is the evolution of one of the protons, of course, and of course the phosphate group, but the reduction there is, of course, the loss of oxygen. So I think that is pretty good, is going to be pretty helpful moving forward. All right, metabolically relevant nucleotides. Now we're going to just switch from ADP and ATP to acetyl coenzyme A and coenzyme A. As these are a larger molecule, and the whole thing is acetyl coenzyme A. In this high energy form, what is shown is the adenosine component that also has a bit of phosphate character. You can see about that, right? It almost looks like it has ADP structure almost, maybe even, I mean, I know we have three phosphate groups. They're not all linked in a chain, but it almost has like that ADP then or ATP kind of formation, that kind of character as one part of its structure. So you know that it's going to be involved in energy transfer. You know that it's basically a type of nucleotide, right? And because you still have the nitrogenous base, you still have the sugar entity, and then you have these phosphate groups, which are a little bit more complicated, especially this one. And what you're getting here is in the middle part, which is shaded, it's pantothenic acid, B5, okay? Vitamin B5 is right here in that lightly shaded area. And where's the acetyl coming from? That's coming from on that left side of that shaded region where you have that carbonyl, right? The C carbonyl, oxygen, right, feature here in that left shaded part. That's also connected. Obviously, we have an amine group there. That is where some of this acetyl group is generating really, and then you have the rest of that molecule. Right, you still have this thioester functional group, and there's also the acetyl component there too. So you can even look at it from that point of view, where you have the carbonyl, but on the left you have that methyl group, and then you have on the right the R group, whatever that is, and that's going to be linked to that sulfur, that you have that C and S sulfur bond, which they consider that to be a thioester. It's an ester. I mean, again, we're seeing the carbonyl. Carbonyl gives you a hint that it's going to be either carboxylic or it's going to be an ester, and because there isn't an OH attached right nearby, then you know that that's going to be an ester, not a carboxylic acid. So you have a couple acetyl characters here. The primary one's going to be the one on the far left, with the methyl group attached to the carbonyl. That in turn is attached to that sulfur. That is going to be the high energy area too, by the way, within the structure. I mean, you know that sulfur is found in the periodic table, right underneath oxygen. You know there's an abundance of electrons present then on the sulfur. You have a couple lone pair, just like you have a couple lone pair on the oxygen. Plus you have a carbonyl area there too. That is going to give us the active area. Okay, so I tried to make a note of that here. The high energy is derived from that carbon sulfur, okay, from that thioester functional group. And this is an important enzyme. I mean, we even talked about this way back with the acetylcholine, right? And nerve transmission, right, of signal via the calcium ions, for instance, or the sodium ions, using the neurons with their dendritic compartments that we talked about several chapters previously. Acetylcholine is synthesized in part by the action of the acetyl coenzyme A being present, right? So this is a metabolite. I mean, this is so important, right? It's using the catabolism then of proteins, carbs, and lipids, at least carbs with the aerobic, in an anaerobic environment. The low energy form then, well, you lose that acetyl part, and that's where you have the acetyl coenzyme A in the upper cartoon, but in this lower right-hand cartoon, you don't. You just have it as the coenzyme A, okay? Because that acetyl group, that primary acetyl group that was there on the left, now it's just as a hydrogen attached to the sulfur. So now you just have your thiol group there, and then that's it. And so that's just what's considered to be a low energy form of this. Now, I'm going to make a note here that in addition to citrate, coenzyme A is produced in step 1 of the citric acid cycle. We're going to talk about the citric acid cycle and the different steps in it, but it's important to know that, citrate is there, but coenzyme A is also there. So we'll be revisiting this, and it's also produced when succinic CoA is hydrolyzed in step 5. So it's going to be a major part of that transfer of energy, from a glucose. You start with the glucose, and how much ATP energy do you derive from one molecule of glucose? We will be answering that but as part of that process, it's a 10-step process, the coenzyme A makes itself known. All right, then we have nicotinamide, adenine dinucleotide, and then you have its oxidized form. So you have the reduced and the oxidized form. The reduced form has the H, and then the oxidized has the N+. Notice that you still have that nucleotide structure where you have the nitrogenous base, you have that sugar set, you have some phosphate groups, but instead of that third phosphate, right, the phosphorus is just, well, that phosphate is connected to another sugar, which is connected to another type of nitrogenous base. So we're considering that to be the nicotinamide, right? That's B3, also known as. So this is pretty interesting, right? These are coenzymes, just like how you have cofactors, I guess, if you want to refer to it like that. I usually reserve the cofactors with respect to maybe the metal ions like magnesium or something, but it's not inaccurate to say that these are cofactors as well. So, and these are going to be the agents that are going to be responsible for helping keeping the balance of the electrons. All right. So NADH, the high energy form, and the low energy form is the oxidized form. Okay. So they are always present in the redox pairing. For instance, if there is going to be a reduction, let's say a TP going to a DP, well, NADH and NAD plus may be present. If we're going from, I don't know, maybe a one type of carboxylate to another type of carboxylate, and I'll show you an example of that soon, you're going to see that that these nucleotides may also be present. Again, it's to help keep track of the electron movement. I think that's the biggest take home from what these enzymes represent and stuff. So, but let's take a look. Under anaerobic conditions, and if you have pyruvate, for instance, we're going to be visiting this, in fact, pyruvate, which is generated in the first step of glycolysis, actually, with respect to NADH, and as long as there is a bit of an acidic environment, and the reaction of that, you're going to have pyruvate with carboxylate going to ethanol, and that's going to involve some carbon dioxide, and then for the charge balancing, to complete the rest of that redox, you're going to have NADH on the left, and then you'll have NAD plus on the right. What is this going to be commonly found in? Well, anything that yeast is fermenting. So I have a demo that I'll be talking to you guys a little bit about where this is in case the fact, where you can make, for instance, alcohol from wine, or from grapes. Got the wrong thing there. And here you have pyruvate actually going to lactate. So depending on whether the environment is going to be anaerobic or aerobic, that can dictate things, and it also dictates what types of cells that we're talking about, right? So you have yeast, or you have yeast cells obviously differ from our own cells, our muscle cells that we have in our bodies. So in our bodies, in our muscles, pyruvate mixed with NADH is actually going to produce lactate, and it's one of the reasons why some people say after you exercise it gets sore and you got to get the lactic acid out. Well, it's technically it's called lactate, but it's produced because in your muscle there's not much oxygen. There's a lot of transfer. If there is any CO2, it's being taken out, right, by the hemoglobin and whatnot, and oxygen being delivered, the whole bit. But in the absence of oxygen, and as your muscle continues to to build and stretch a little bit, right, due to that exercise, right, due to that stressor, then it's perfectly normal for lactate to be produced. So different kind of products that can be formed then in an anaerobic environment, especially if you're considering yeast versus aggressive muscular exercise. So whenever we aggressively exercise, we're not actually producing ethanol, unlike the action of yeast, which loves to gobble up some glucose in particular and convert it to pyruvate, which then quickly, relatively speaking, converts to ethanol. Under aerobic conditions, you still have oxidative decarboxylation of pyruvate. Okay, this is within the mitochondria. And these groups are shaded to try to help show where the action is coming from and going. So in this particular case, you have coenzyme A, okay, in its Well, in its low energy form, right? Because this is just the thiol group capped onto the coenzyme A, as opposed on the left here, as opposed on the right, which is in the purple and yellow, you see the acetyl coenzyme A. And the acetyl coenzyme A, again, is the high energy form. And so there is an exchange that happens. The acetyl group is actually derived then from part of the pyruvate molecule. So part of that splits, it reacts with the coenzyme A to form acetyl coenzyme A. In addition, you're also going to liberate some carbon dioxide and NADH. The high energy form is going to be produced from that NAD plus low energy form. So these are just all examples. And again, why they're there is to try to balance the redox action going on, right? And whether you're gaining hydrogen or losing hydrogen, or you could even, consider that to be electrons instead, but it's NADH is quite nice, and NAD plus with respect to just keeping track of just where the hydrogens are. And this, you can see whether it's aerobic, anaerobic, yeast, muscle cells, or even under aerobic conditions, where all of our cells, not just our muscle cells, of course, are undergoing some kind of stress. You can easily see how the importance of having coenzyme A already in our bodies, a reservoir of that, as well as if you have energy already, available or not. And for instance, glucose or glycogen, maybe it's eventually broken down into pyruvate, for instance, and then reacted with coenzyme A to give us that energy to allow us to do the vigorous exercise. I mean, that's just what goes on. But that is, this process here is what's going on in the mitochondria. So it's all part of glycolysis. And we will be talking about that shortly. I keep saying that, but it's going to happen. Just wanted to introduce a little bit more with respect to some of these nucleotides because they are so important. Here's an example then of NADH and NAD+, right? You have reduced form and an oxidized form, and this is the one step of the citric acid cycle, as I mentioned. And this is during the conversion of isocitrate to alpha ketoglutarate. Alpha ketoglutarate is one of the alpha keto acids that is commonly produced perhaps from the processing of fatty acids in the body. Again, we'll be talking about that, but this is also just one of the steps that is native to the citric acid cycle. Isocitrate, as shown here in the cartoon on the left-hand side, is converted to this alpha ketoglutarate, but it can't be done without the presence of NAD+, which of course, then gets reduced to the NADH. So it's really important that this dinucleotide, each one of these are present. And so I try to show this, where's that redox balancing from? Well, you have isocitrate that has actually 3 charges on it. You can look at 1, 2, 3, just count them, right? And then that goes to alpha ketoglutarate. Well, how many charges does it have? It only has 2. So it lost 1, got oxidized. The NAD+, well, it was present. You can see how in the cartoon here under the number 3 on the left, you can see how it can take in a hydrogen and then, lead to its reduced form. Again, in that reduced form, that doesn't mean that it's inactive. In fact, it's the high energy form. So that reduction is actually going to help kickstart other processes. And so with the evolution of carbon dioxide, which needs to be, of course, cleared from the cell, and then, of course, the body, you also have to clear out this high energy form, but at least it's there. It's going to help mitigate some of the other processes that are going to continue. So this is only step 3 in the citric acid cycle, and there's additional steps. And so that's where, it helps to have this cycle continue, and it shows you that this is not the end of the conversion of, for instance, glucose all the way to energy. So this is just one step in it. And you do have, the generation of a high form of this dinucleotide, but its form is as a way to balance the electron transfer and mitigate heat evolution. So all that is important. You also have flavin-adenine dinucleotide, okay, FADH2 and FAD. All right, so you have riboflavin. You guys are probably familiar with this as one of the vitamins as well, just like how you already had the other vitamin, B3 or pantothenic acid. And of course, you had the nicotinamide. I mean, all these things, right? All these things are vital vitamins, and you can see, you can understand how then having these nutrients in our bodies are so important because these are going to be involved as coenzymes in all of the different processes of using some substrate, some relatively simple molecule, i.e. glucose, and converting that to something that can be used and derive us energy for which we can do things. So in this particular case, again, you have your reduced form, then you have your oxidized form. The reduced form with the FADH2 is going to be your high energy form, okay? It has more hydrogen, so that's going to be involved in some stuff. You still got the nitrogenous bases, you still got the cyclic sugar component, right? You still got the phosphate groups, and then you have this other substitution here, which has, all of these other different types of, it has a nice carbon chain, and then it has this other type of nitrogenous base, which is, you have 3 cyclic groups here all bonded together. And so you can see where the H's are coming from. They're all attached to the nitrogens. And if nitrogen is going to lose its hydrogen, within this riboflavin kind of environment, then you're going to get into that low energy form. So whenever you're considering, well, geez, where are the hydrogens coming and going? Look at the riboflavin. And in particular around the cyclic structures where we have the hydrogens attached to those amine groups, okay? So here would be an example of the presence of FADH2 and FAD in, again, the citric acid cycle. This would be in step 6 of the citric acid cycle. So in this particular case, you have succinate, okay, which is shown on the bottom of this cartoon, converted to fumarate, all right? And fumarate has only 1, 2, 3, 4 carbons. Here, the succinate has 1, 2, 3, 4 carbons as well. Interestingly, although they both are 4 carbons, you can tell that fumarate lacks some hydrogen because of that presence of the double bond. So that fumarate structure is going to be obviously involved in some of the balance, but it's going to be a more oxidized form because it doesn't have as many hydrogens, right? So that's what you have to look at here. What does FAD then participate in? Like, what does that look like? Well, it gets reduced because it takes those 2 hydrogens from succinate and it attaches them onto itself, right? It becomes a high energy form and you get FADH2. Additionally, succinate, as I just said, becomes oxidized to fumarate, okay? So this is just, again, another process in the citric acid cycle. You're gonna have all these little carboxylic acids, well, the carboxylates really, because that form dominates, with respect to our physiological pH, especially that within the cytosol, right? But all of this is about balancing and trying to mitigate the evolution or loss of electrons or hydrogens, really. I mean, but all this stuff has implications for ATP. And so these units here, every time you see, for instance, FAD going to FADH2 or NAD plus going to NADH, all that has a characteristic ATP, quote unquote, energy level. And that's worth something. And I'll show you what that is worth again in a moment, but know that that's going to be a conversion to be like, well, you keep on saying ATP and we're going to be looking at the citric acid cycle, but if we're talking about FAD or NAD, how does that translate? Well, I'll show you. There's actually like a conversion that one can use to say, okay, if I have NADH 2 or NADH or FADH2 present, then I know how much ATP energy that is worth, okay? So it'll be a conversion. It's just like it's a conversion from going from atmospheres to pounds per square inch to millimeter mercury or something, okay? So there's just a bit of a conversion like that. All right, so here's the example then of electron transport involving both of these dinucleotide combos, okay? So you have the NADs and you also have the FADs, okay? The flavins and the nicotinamides. And here, this is going to all basically occur, I don't want to say, simultaneously, but it is going to be pretty close, okay? So NADH dehydrogenase, okay? If you see the ACE, that's going to be a protein, and that's going to be an enzyme at that. It's going to be at this what's called complex 1, which is going to be another type of protein. And I say that this is going to reduce ketones of quinone to alcohols. So let's take a look at what I mean by that. So here you have the phospholipid bilayer, okay, characteristic of cells. So we're on the cell membrane, and we have this complex one. So this is pretty new, right? So this is new, and this is probably going to be, is this, well, first of all, what type of membrane? This would probably be within the mitochondria, right? Because this is where energy is being produced. So we're not talking about the outer limits of the cell, like as a whole, right? The whole eukaryotic cell. We're talking about only one part of that organelle. And at that, within that boundary layer, you have the phospholipids, and you have this complex one, right? And then you have an imbalance or separation of charges. Let's just say there's positive charges on the outside of the cell, and then you have the negative charges inside. Again, we're talking about the mitochondria. If NADH is present, okay, it can become oxidized, right, to NAD plus, and in doing so, the H plus, the proton or the electron, they're going to be attracted, and then it's just going to pass on through. So the NADH plus, and in an acidic environment anyway, H plus this Q, this quinone, will then undergo the redox reaction where NADH plus is formed plus this QH2. Okay, so one's going to get oxidized, one's going to get reduced. We understand that. And what we're really talking about here is, at complex one, within this complex one is where this quinone, okay, is undergoing this reduction, right? You see the oxidized coenzyme Q here, where you have these ketone components, right? These are the ketone functional groups in this cartoon on the left-hand side. In the presence of a couple protons, right, which is balanced by the 2 electrons, then that can readily get reduced to alcohol. So then you got hydroquinone. So you go from quinone to hydroquinone. So that is an example of what's going on, for instance, at that complex one, which is a protein, right? It's a massive protein, and it's facilitated by this NADH dehydrogenase. Okay, so it can't just do that all by itself. And then you have succinate dehydrogenase at complex 2. So this would again be another example. Here you have Q, shown within this lipid bilayer, right? You have this different type of complex 2. It's way different than this complex 1, but it's helpful in showing that, for this particular case, that you're reducing ketones. I mean, that is the main thing here. FADH2 is presented, and it's able to interact and reduce this quinone. Okay, so this would be, for instance, coenzyme Q shuttling electrons along my mitochondrial membrane during the citric acid cycle. Reactions enrich to water, en route to water and ATP formation. So these are just examples. You have to memorize this? Oh, no, you don't. But this is just an example of where this FAD and the NADH are active. They're present within the mitochondria, and they are so important in being able to reduce ketones. Okay, and in response to that, all that action is the eventual evolution of, water is going to be a byproduct, of course, but then, of course, energy formation, okay, ATP. And so that's the energy currency in which this is being used. So it's complex. This is not easy stuff, and could you imagine trying to keep track of all that, on a really tiny scale? So all these scientists and researchers who have been studying this stuff for years, I mean, hats off, right? Because all these different molecules and such, they have more than one purpose, and that's not an easy thing to suss out whenever you're trying to figure out, well, what's going on. All right, so now we can start talking about the digestion of different foods, and let's start with carbs. So really, digestion of food, mostly it's going to depend on what type of organ, but with respect to carbs, it begins in the mouth, okay? Because we have, in our saliva, we secrete these, for instance, these alpha amylase. Amylase is really important, right? I mean, that's one of the enzymes that is found in, for instance, BNO that could help break down some of the polysaccharides. So it's decent at breaking down starch, but not all starches. And, for instance, we can't just chew on straw like a cow does. I mean, we already talked about that previously. But, there are limitations to our to the power of our enzymes that are secreted, by the glands, in our oral environment. So anyway, digestion of carbs begins in the mouth, okay? And then it proceeds where if it can be broken down into smaller components in the stomach, then it will. But if it can't, that's fine, because in the small intestine, there are enzymes present that can help break these things down. For instance, lactase can break lactose. Maltase can break maltose. Okay, even in the pancreas, there's a bit of that too. And of course, sucrase. So sucrase is also secreted. So the small intestine is so important, right? Lactase, maltase, sucrase. This is where all these enzymes are going to be useful in breaking down some of these more simpler carbohydrates. These smaller saccharide molecules are going to be. And then of course they will be taken and absorbed into the bloodstream. Okay, everything still may pass through the large intestine, but that's for absorption and then passage into the body. The small intestine can also have absorption, through itself and then into the bloodstream. So the large intestine isn't necessarily critical for that, but it is definitely helpful for further absorption of those nutrients. So if you can get absorption in the body from the small intestine, that's one area. But if whatever isn't absorbed, into the body from the small intestine, that can pass to the large intestine and then also be absorbed into the body, then that's another benefit. So that's one of the advantages of having both your large and small intestine, right? We take it for granted, but there's a lot of people that struggle with that, or that may not have, both of those organs. All right, so digestion of lipids is another one. Now here, digestion of lipids really begins in the small intestine. So notice that this really isn't beginning in the mouth. We don't have those kinds of lipolysis enzymes that our saliva secretes. And if we do, it's not to any appreciable degree. So this has to be all the way down and into the small intestine before the digestion of lipids can occur. And then of course, it goes into the large intestine as well. So begins in the small intestine and just carries on through to the large intestine, where again, other enzymes are present and they can help aid further digestion of this, and then of course absorption. But these are just examples of this. I mean, even in this pancreatic lipase, when this lipase again is also present in the large intestine too. Okay, it's not just within the small intestine. So the pancreas helps, the small intestine helps, the large intestine helps. All of this stuff helps. All right, but fats can be processed in the lower part of the digestive tract. And you already know from the condensation reactions and the hydrolysis reactions, right, that you're going to have some triglycerides, right, that are going to be formed. The triglycerides are formed into smaller units by action of the enzymes that can help break that down. Usually these are, larger chains, and that's kind of what this upper cartoon shows, is that you have the one, polar head group, and then you have this long tail, right? Well, that's like fat, but that can get busted up by the lipase, right? And then you can have additional absorption of that. So here you have a cholesterol, you have a fatty acid tail. Guess what? You can form an ester, right? Based on this, we haven't really talked a whole lot about forming esters like this we have, but not recently, right? Where you have an alcohol. What's the combination? What's the reaction, the synthesis if you have an alcohol plus a carboxylic acid? It's an ester. But we haven't talked necessarily about a cholesterol type of ester. But that's really what this is. So if you're talking about any kind of cholesterol, these can be larger molecules for sure, but they don't have to be. So even glycerol would be an example of this type of alcohol group, right? But again, you wouldn't think of glycerol as the typical cholesterol type of molecule. Usually that's a bit more larger in size. So, but the whole point is all that uptake is there. Whatever isn't taken up in the small intestine, again, is going to be passed through the large intestine. There's going to be additional enzymes that are present, and major absorption occurs. And it's actually, one of the most important ways of absorbing water and salts and stuff into your body is through the large intestine. So I think that's pretty important. Now you will get some bile salts that are going to be there. Here this shows that the catabolism of large non-polar globules in the micelles and small droplets occurs within this area, and that is true. I mean, again, the small intestine is kind of the balance between this very acidic stomach and the relatively neutral large intestine. So, you know, you're going to have these kinds of phase mixing, really, if you will. So you're going to have these non-polar globules. You're going to have these, or droplets, as I put in parentheses here. You're going to have some of these different types of small molecules or different mixed phases, and all that is going to be handled uniquely in the large intestine. Okay. So esters and proteins transported via the bloodstream. All right. Now, with respect to digestion of proteins, right, you have your stomach, right, that tries to denature the protein. How does that happen? It's an acidic environment. So if you go back and look at some of those demos, I didn't have to use heat to denature milk or eggs, right? Those are two classic examples where I did not have to use heat to denature those proteins. We can do so using an acidic environment. And that's what you're seeing here in this first part. So you look at the stomach, you see the proteins, right? It's made up of all these amino acid spheres, right? They have all their tertiary structure, but then you need to unfold that tertiary structure. And that's what, again, part of that acidic environment in the stomach is doing. All right. And then the small intestine introduces, again, some different types of enzymes that can further break that down, right? You have this pepsin or chymotrypsin, or even just trypsin. And pepsin is also present in the stomach as well, too, sometimes. So, so it's still there, but what's that doing is it's busting up the protein, getting into the amino acids so that they can be anabolized and used by the various tissues for whatever purpose. Okay, so digestion of proteins, again, not really in the mouth, but definitely in the stomach, okay? Uptake via small intestine. And that is what we're doing there. So we have now the background to start talking about the glycolysis and what I call them 10 glycolysis commandments. All right, and the first 5 steps are all going to be endergonic, meaning they're going to use energy. They're going to use up this ATP, any kind of ATP currency or ATP-like currency that is going to be available, is going to be necessary for actually getting this feedstock into a manageable form where it can then proceed to give us energy, you know, produced instead of us, you know, taking it. So in the first part, right, of glycolysis, you're going to start with glucose, okay? And if you don't have glucose, then you haven't digested as well. Remember, sucrase helps to digest sucrose, and that happens in the small intestine. I shared with you a couple slides back. So whenever sucrase is going to do its action, now all of a sudden glucose is a smaller entity. Now it just got absorbed right from the small, maybe even into the large intestine, you know, from there into our bloodstream. Now what's going to happen? Now the cells can start using that glucose to do something, to convert it to energy. And so this would be the start of it. So you have ATP, right? Ooh, look at that. It gets oxidized to ADP. I should say, reduced, right? So ATP gets reduced, glucose then becomes glucose 6-phosphate, okay? So that gets oxidized. And so you can see how the process will just continue to unfold. So you have 1, 2, 3, 4, 5 steps here. We're going to talk about those in a little bit more detail, but importantly, regulation of glycolysis is dominated by step 3, okay? We have this fructose 1,6-bisphosphate, okay? Here you have phosphofructokinase catalyzing the phosphorylation of fructose 6-phosphate, which sounds, well, obviously very wordy, but this is where the action is gonna happen in terms of the regulation of this. And I'll show you what I mean by that in just a minute. We'll be talking about it, but this is the most important step right here in terms of how much glycolysis is going to be needed. And what happens if you don't have glucose, for instance, admitted into this cell, but you have another type of sugar. So that's what I want to talk about in just a minute. So then you have this dihydroxyacetone phosphate, you know, that gets converted to this glyceraldehyde 3-phosphate. And that is, you know, right before that happens at step 4, aldolase, another enzyme, is splitting that bisphosphate, okay, into 2 triose phosphates, all right? And you can see how they're how that in that cartoon there on the right hand side where it branches into a right and left arm, although they have different names, you know, you can still see that there's still three, right? You still have three carbon entities, right, on each one, but the phosphate is just attached, you know, on either side, but it's a way that those carbon entities are attached, which makes this different, okay? But this is the most important part, because this is where one glucose molecule that you started with here at the top of this glycolysis process produced two pyruvate molecules. That is the most important part, okay, of that in terms of knowing, okay, what's going on? We're starting with one, now we got two. That pyruvate will then subsequently be emitted for the rest of the cycle. You'll see. The second 5 steps are going to be exergonic, and these are the rest of those. So this is where in step 6 you have an NAD plus coenzyme. All of a sudden, right, the low energy form, the oxidized form of this coenzyme starts to be, you know, starts to make its presence felt. So you're going from an oxidized state to a reduced state, a high energy state, just like how upstairs here in the first 5, you started with a high energy ATP and you went to a low energy ADP, right? So as soon as step 5 has been completed, and then the rest of the 5 steps can be completed. Now you're seeing how instead of going from, you know, from a oxidized to reduced, okay, which is what is going on right here in the latter half of these 5 steps, you know, in the top, you went from a reduced to oxidized. So reduced oxidized, first 5, now you go oxidized, reduced. In doing so, ATP is produced, okay? So let's look at this a little bit more. And before we do, actually, this is where I want to talk about fructose. Fructose as an energy feedstock, right? This is really interesting, and this bears on fructose corn syrup, whether it's high fructose corn syrup or not, but this is pretty interesting. Fructose is immediately directed to the liver, okay, you know, following step 5 mechanism, where it's catabolized into treosis and muscle tissue, okay, is converted to fructose 6-phosphate like glucose. But the important thing is here, unlike glucose regulation, okay, which is dominated by this 3rd step, okay, there is no regulation of fructose. There is no enzymatic action. There is no, we don't have the proteins that will signify, hey, look, we need to stop producing more glucose, fructose in the body. And that is where some of the complications can thus arise. So just like how you have the glycogen with the insulin and stuff, and that can help regulate how much glucose is being produced and or emitted into a cell, right? Fructose has no insulin-like analog that will help limit its admittance into a cell and or its production even. So if you can see that if there's no regulation of this fructose, then there's probably going to be a buildup, and it's going to be a buildup in one of the organs in the body. Is definitely going to be a liver, and this is what is going to start giving rise to compromised metabolic action in your body. And so if you hear reports, if you hear scientists say, look, you know, fructose is having this kind of damaging effect on the body, well, it's because there is no regulation of this. We don't have the proteins that can adequately handle fructose, not only production, but then use, right? Basically regulation. So it's pretty interesting. And once that happens, then you have, like, for instance, as shown in this cartoon right here. So you have all this on the left-hand side. You have this, you know, the reaction steps. I told you about the 10 commandments of glycolysis, right? You start with 1, you go all the way down to 10. Regulation step is number 3. So that's going to be the bottleneck if we were to have like a flask. And we were to say, okay, well, we know glucose is regulated through glycolysis, but what about fructose? That's the problem. If you have too much fructose that's being entered into your body via the diet, I think that's really important, okay? If it's being introduced into the diet, this is where the problem occurs, because it will enter glycolysis, but then it'll just keep going, and then all of a sudden you'll have excess product, and if you have too much of that, it'll be converted into fat. And this is where, if it aggregates and collects, and this is where the metabolic syndrome or metabolic panel can start to suffer, within, one's body. And, that is itself the damaging, scenario. So just know that. So fructose is not regulated. There is no insulin-like analog for it. If you have all this excess, fructose being presented, well, then it's going to eventually aggregate and be converted to fat because it can't be consumed. Pyruvate and acetyl coenzyme A. Now we're back at these guys again. Okay, so where did the pyruvate generate? That was in step 5. It was right there in, or right, right amongst that. It was within that first 5 steps of the whole glycolysis process. But now we're going to be looking at it with respect to, well, do you have oxygen present or do you not have oxygen present? If you don't have oxygen present, then whenever glucose gets to pyruvate, right? And that was, let's go back a little bit. Actually, I will show you to help make this a little bit easier, right? This was in right before you get to step 3. Well, it's right in step 4. You see here where you have aldolase splitting the fructose. Okay, that was regulating step 3. This is where the 1-glucose now gets into 2 pyruvate molecules. Okay, so we're looking at step 4, and step 4 is then going to go, to step 5, and then it's just going to continue on. So I think that this is important. So step 4 is where this is happening, and where pyruvate is generated. Remember, for every 1 glucose molecule, you're getting 2 of the pyruvate molecules. We're not showing you 2 pyruvate molecules here. We're just showing 1. But the point is, you have either an anaerobic condition or you have an aerobic condition. The cytosol has an anaerobic environment, whereas in the mitochondria, it's an aerobic environment. Okay, and in the cytosol, I want you to think about muscle tissue. Okay, so if you're thinking about muscle tissue and you're exercising and all that other stuff, this is why you get sore, lactate is produced, right? You have NADH, okay, it gets oxidized to NAD+, okay, low energy form, and then you have lactate being evolved here, and lactate is being reduced because you don't have that, you don't have 2 carbonyls anymore, you only have 1 carbonyl, okay? So one of those carbonyls turned into an alcohol. All right, so you have reduction there, you have oxidation, you have reduction with the lactate, right? And you have oxidation here going to the NAD+. If you're looking at from an aerobic environment within the mitochondria, you have these coenzyme A's, right? You have the low energy form, which is the HS form. You can consider that to be, I don't know, the oxidized form, I guess. But then you have NAD+, right? That's oxidized, it goes to a reduced form, right? And so now you have the acetyl coenzyme A, which was produced, and that is then a higher energy form of what is going to be needed, and that's what helps basically kickstart that citric acid cycle. Okay, so again, every 1 molecule of glucose produces 2 molecules of pyruvate. This would be the chemical reaction that you would have. Instead of me drawing the arrow from left to right here in the middle, I just have it going from top to bottom just because I'm running out of space. But you have the glucose ring, okay, that is aided by the coenzymes NAD+, and ADP also is present along with phosphates that are hanging around from other processes that are also going on, right? So you have some additional phosphates there. Well, the ADP and phosphate, you can imagine that, and combining to form ATP. That's not a hard stretch, right? So you have that process, plus you have glucose going to 2 moles or 2 molecules of pyruvate, and you have obviously the reduced form here of the NADH as well. Notice that this is still a bit in the acidic environment, okay, because you still have some free hydrogen there, or protons, I mean, you have the carboxylate there. So, even though it still might be acidic environment within physiological pH, which is around 7.4, this is going to be preferred because, well, just because of the nature of that acid pH of pyruvic acid, which is definitely less than the physiological pH. Now, I know that these are a lot of videos. I have 15 videos corresponding to what this is, but I think they're all pretty short. They're all like a minute or so, maybe 2 minutes. But this is where actually I show the power of the yeast and being able to ferment grape juice, which already has sugar in it. There's like, I don't know, about 38 grams of sucrose in a standard serving, which is about 240 mils. I didn't use all 240, but it scales, right? I think I used about 50 mils here. But I use the yeast. I just use this rapid rise, and you can see what those yeast cells look like here, right? You have these little microorganisms. There's about 4.5 grams there in that cup. I added them with the 50 mils of grape juice to these different test tubes. Some of them I had a screw cap on, some of them I just left them open to the environment, and others I put a balloon on. And it was really interesting because, you know, over the course of some time, I mean, there was definitely some action within the first hour, definitely within the first 8 hours, but I let it go all the way to 7 days. But what you're basically relying on is the yeast to be able to use invertase, which it has. It's an enzyme that can break down sucrose to glucose, kind of like sucrase, right? That we use, for instance, in our enzymes. They use invertase to get it to the glucose feedstock. The glucose feedstock then is produced by way of another enzyme that it uses to conduct the glycolysis to 2 pyruvate molecules. So you already know that from what we just talked about. And then those pyruvate molecules, whenever it is combined with one of the dinucleotides, right, with one, or the NADH, right, one of the coenzymes, not dinucleotide, but just a regular nucleotide, we have a redox reaction with that pyruvate plus the NADH. Well, ethanol is produced as well as carbon dioxide gas, which is just what we said in the previous slides. The carbon dioxide, guess what? That's marked by the evolution of an inflated balloon, because it's able to, the gas is so strongly able to be produced that it's able to push back on the atmospheric conditions, and you're able to inflate, and you have this process then being able to occur. So by virtue of just the inflation of the balloons, you can know that ethanol is being produced. So pretty neat, because you can't get the carbon dioxide without the ethanol. But this reaction from pyruvate directly to ethanol is going to be pretty quick. All right, and it's going to be pretty quick because NADH is present within the yeast, and it's, you know, within the mitochondria there of the yeast, and it's going to want to get this action going. And you know, ethanol and carbon dioxide are going to be the products of that. So, and then I looked at what the difference in the top fluid versus bottom mixture would be, and it shows that in the bottom mixture, basically where there's a higher concentration of yeast, you're also going to have a higher concentration of ethanol in this particular case. And then again, I didn't mix these up, I just let it set. There could probably be an even better example of this if it was continually agitated, but maybe not. But some of the alcohol does evolve and get into the top liquid layer. Again, what do I mean by that? Just go to the videos, you'll see, read the descriptions, and you'll be able to better understand. But I use these saliva tests, and a darker test on the tops of these test strips indicate that there's going to be a higher concentration of alcohol then, as going from left to right, from white here to this like navy blue to the right hand side of that on that test strip. And you could easily see that in the bottom mixture where there is yeast, you could definitely see how the tops of those test strips appear darker. Well, and it's because there's more alcohol present. So it's just an example of it. You could do it with grape juice, and you could add other things to, like if you wanted to add additional sugar to it, you can do that. If you wanted to, and I have a whole separate video, I haven't posted it yet, but I could. If you didn't even want to use yeast, if you just wanted to ferment a banana in a thermos, you could just use whatever was on the banana, whatever bacteria, whatever mold, yeast, fungus, well, I know, you know, whatever it is, whatever automatically just resides on the banana, if you put that in a thermos, it will ferment alcohol, okay? And moreover, if you were to add some sugar to that banana and to that thermos and just let it sit there, again, add a little bit of water to help make things mix up, then you can also produce some ethanol. Again, almost on par here with what the yeast is doing to grape juice. And that is actually one of the ways that inmates make what I call jailhouse hooch, because they're unable to get the alcohol as easily or readily. Usually that's the case, so they can make their own. Anyway, let's talk a little bit more about the citric acid detail, because this demo and stuff is all about this. You know, at least the first 5 steps where we're actually creating alcohol. So we got the first 5 steps are energy intensive, meaning it's gonna cost, and then the last 5 steps are gonna be energy producing. So let's go over these. So you have the formation of the citrate, and well, that's why it's called, the citric acid cycle. It can also be called other names, but you have all of the rest of the cycle. So you already knew that you had some of the pyruvate, right? It breaks up, some of it's gonna interact with coenzyme A. It's gonna form this high energy form, right? This is gonna be under aerobic conditions. This is gonna occur in the mitochondria. Okay, this is where the citrate is going to be produced, okay? You have number 2 going right here. So you have the acetyl coenzyme A has now been introduced right into the cycle. Now you have citrate. Citrate is there on the right-hand side within that shaded, or I should say, dotted or hashed black box, right? You have the citrate molecule. Again, citrate is no different than what we had already done with citric acid, whether we were making those kind of starch tart things or whether we're talking about the basic carboxylic acid, right? It's all citrate. It's the same general form there, okay? We have 1, 2, 3 COO groups, okay? And that's consistent with citric acid having those, right? The 3 carboxylic acid groups. So you're gonna have this isomerization going from citrate to isocitrate, okay? And that then, once it gets into isocitrate, is going to lead to the evolution of CO2 and NADH. Remember, NADH is high energy stuff. So this oxidative decarboxylation occurs, okay? And that's an important part in this process because, again, you have the CO2 being generated and the NADH. The evolution of this NADH is going to help kickstart other things and keep stuff going. That's gonna have to be regulated in terms of its energy. You can't just hang out there in that part. That wouldn't be thermodynamically favorable, or it could lead to some unwanted, undesirable effects within the mitochondria as it's processing this. So that in and of itself, once it gets to that high energy phase there in number 3, right, it's gonna kickstart another one, a second oxidative decarboxylation. Okay, so again, more of this carboxylic acid character is being reduced. I mean, you can see that here. If you go, let's go back up to citrate and isocitrate. You see that there's no difference. You still have 3 carboxylic acid groups, right? You have 2 in blue and 1 in purple. But look at what happened whenever you went from isocitrate then to alpha ketoglutarate. In the step 3, you lost one of the carboxylic acid groups. You lost one of the blue groups, you see? And that is where this oxidative decarboxylation process terminology arrives from. Where did that carboxylic acid go? Well, it lost part of it to carbon dioxide. And then the other part, it said, look, we need to, conserve this redox process of what's going on. So NADH was formed, this high energy form, okay? And if we're talking about the second decarboxylation occurring, then at step 4, then you're looking at this alpha ketoglutarate molecule, which still has 2 carboxylic groups. It's now going to be processed to the succinyl coenzyme A. Notice that the coenzyme A, succinyl coenzyme A only has 1 carboxylate group in that purple. So you had a blue and a purple in the alpha ketoglutarate, okay? Undergoing some processes here, which is going to include some redox because, again, you have NADH being formed, high energy formed. You also are going to lose that carboxylate in the blue, for instance. It's going to form CO2, and then you're only going to be left with 1 carboxylate group. So once you get to step 5, you only have 1 carboxylate left. You have the succinyl CoA, but this is still a high energy form because it's still, it's not acetyl CoA, but it's succinyl CoA, and you still have that ester bond right there. You have this carbon sulfur bond, and that's high energy stuff. So you still have a high energy situation in addition to having the NADH, which is also high energy, okay? So you have all of this high energy producing, substrates that is still just continuing to just propagate this cycle on and on and on. Now you get to 5 and you have some hydrolysis of that succinyl CoA. Okay, in doing so, succinate will then be created. And then you also have the evolution of GTP, okay? That has implications then for ADP evolution, all right? Because there's this whole cycle with that evolution of the triphosphate, the GTP molecule. Once you get to number 5 and you have that succinate, and again, it's a redox thing. You can see that. You can see how you go from a thiol attached to the coenzyme A. That wasn't the case whenever you had the succinyl CoA. So you can see that how there's additional redox stuff there. You're going to have dehydration then of the succinate, and in doing so, you're going to lead to that formation of fumarate, which has the double bond. So you know that H's are going to be popping off. That's where you have the FADH plus or 2 forming from that base FAD, low energy molecule. And so it just continues to go. And then you have the hydration of the fumarate, because that's going to open up, right, that double bond. And you still have 1, 2, 3, 4 carbons. I mean, the carbons didn't change as you go from fumarate to malate. But now you opened up, you still have the carboxylates there, but you have a reduced environment, right, that has the alcohol groups, right? And then you have the oxidation of malate, and subsequently that will lead to the evolution of NADH. And then the whole cycle just continues again. So the whole point is you're generating energy in the cycle, and you can see how one goes to another to another. This is basically the summary of the citric acid cycle. You have the degradation catabolism of metabolite acetyl coenzyme A. It's described via the citric acid cycle because the first molecule is citrate. I'm using this acetyl CoA here in this particular case. The acetyl CoA, this acetyl group, still marked by these 2 purple spheres, right? The 2 purple spheres are going to be acting as feedstock, and then it's going to be linking up. It will combine with oxaloacetate there on the left, which had already been processed before. And so you're still able to get the citrate. It goes into isocitrate. There's like that isomerization step. After that isomerization step, it goes to this ketoglutarate, right? You have all these processes. NADH is formed. NADH is formed. Guess what? Additional GTP is formed, which has ATP currency, right? You have FADH2, you have NADH. All of these are energy producing. These are high energy nucleotides, okay? NADH, FADH2, GTP, all of those. So all of this in the cycle is leading to higher energies, all right? And that's the important part to know, because we can start now keeping track of how many of these high energy molecules are being produced, because then we can determine how much energy quantitatively is being produced. So you see that each turn of this cycle creates 1 ATP, okay? And it does so because of the GTP, all right? Conversion up here on top, where you had GTP plus ADP is GDP plus ATP, and ADP is plus PI is 1 ATP. I mean, all this stuff can get bananas. But if you start using that kind of currency conversions, I should say the exchange conversions, you can start saying, okay, so I see that we have an ATP. How many NADHs do we have? Okay, let me just count them up. I have 3. How many FADH2s? I have 1. All right, then that's fine. NADH and FDH are also, again, they have those conversions to ATP. So let's figure out how much ATP is totally being produced. So the overall oxidation or catabolism of glucose is this. You have the glucose molecule in the presence of oxygen ultimately lead to CO2, H2O, and energy. This is the energy, the ATP, that we're trying to calculate. So let's talk about glycolysis, okay? Oxidation in the cytoplasm, okay? Basically within the cytosol, right? You have glucose. Ooh, look at that. It's undergoing 2-pyruvate, right? And you have ATP and you have 2-NADH, okay? So that got oxidized, right? And so then what happened? Well, in the mitochondria, the preferred metabolite is the acetyl CoA, okay? So within the mitochondria, you have 2-pyruvate being converted to acetyl-CoA plus 2 NADH. Why do all those, coenzymes happen? It just balances the redox. That's it. But that's an important part. Again, they help to balance electrons. It's an important part of being able to use those substrates for something else. You have the oxidation of the metabolite to generate more energy via the citric acid cycle, and it's 2 turns because there's 2 metabolites. So from every 1 glucose, you see 2 pyruvate or 2 acetyl coenzymes, right? So what does that look like? You have 2 acetyl coenzymes, then are going to produce 6 NADH, 2 FADH, and 2 ATPs. Where did I get this from? Because each one of those acetyl coenzyme A produces 3 NADH, 1 FADH2, and 1 ATP. So basically just what we had on the previous slide. So then glucose will then just add all these up. So you have the glycolysis, you have the oxidation here, right? In order to get it into the right metabolite. And how is that even possible? Well, because there's oxygen in the mitochondria. It's an aerobic environment, all right? And then you have the remainder of the citric acid cycle being converted, that acetyl-CoA to something more useful to us, i.e. the energy. So you have glucose, 1 mole of glucose, then just add up A, B, and C totals. You have 10 NADH, 2 FADH2, and 4 ATP. Accepted conversion, this is what I alluded to earlier. NADH is about 2.5 ATP, and FADH2 is about 1.5 ATP. That's the energy conversions, okay? So that's how you get your 32 ATP. So 32 units or 32 ATP units are produced from 1 molecule of glucose. That's the catabolism, okay? And usually there's another conversion that you can use too, where acetyl coenzyme A gives rise to about 10 ATP, so that's another one. This is a visual summary if you want to, and here's a table 12.4, tries to give you some of those different conversion units. Gives it a little bit different with respect to how it describes the ATP yield versus which pathway that they're talking about. So this might be helpful to you. You can look at that. But this is the overall schematic, again, where you have glucose, right? And it gets converted to pyruvate, and then pyruvate gets converted to acetyl coenzyme A. Again, why 2 acetyl coenzyme A and not lactate? Because it has an oxygen-rich environment, okay? And because it is oxygen rich, this is why the whole cycle goes the way it goes. And so all of this tries to keep track of the different energy evolutions that are happening at different parts of that cycle. So pretty interesting. All right, so we're doing pretty good. We're almost through it. We got to get through the catabolism of fatty acids now. And because fatty acids can be used for energy. So, and how does this even happen? Well, first, there's always going to be hydrolysis of the glycogen, okay? Generally, they call that glycolysis. You have gluconeogenesis from proteins and triglycerides when glycogen is depleted. This is a very common situation. So anybody who's starving themselves of carbohydrates and wants to try to burn more fat, for instance, if you're on a keto diet, this is something where your attention would be directed to this. Or if you're just, you don't even have to be on a so-called keto diet, just be on a low carb diet. Well, what we're going to be looking at here is instead of an acetyl, we're going to be talking about an acyl coenzyme A, okay? And really this is fatty acid, and we're going to use terminology that talks about where these reactions are occurring. So we have our alpha carbon and we have our beta carbon, okay? The beta carbon is going to be split. So the way that this pathway proceeds with the catabolism of fatty acids is it starts to split up units. So you have 2 carbons per quote unquote pass that form an quote unquote activated fatty acid. This is just what has been observed. So you might have this really long chain. As long as you have this coenzyme A present, as long as there's an abundance of this coenzyme A present, it's going to split that fatty chain into groups of 2 molecules, okay? And here's a cartoon and how it does it, all right? And it's gonna then, if it's gonna do that, then you're gonna have to invoke a little bit more of a coenzyme kind of cooperation to be able to make this happen. And so FAD and NAD are both shown to be present during the catabolism of these fatty acids. So you have the coenzyme A, it's attached to the sulfur group, right? So you already know that this is a high energy form. Okay, you have FAD, oxidized low energy, going to FADH2, which is a reduced and high energy form. We have the beta carbon. That's going to be where the split is going to occur, where you're going to get these multiple units of these two carbon species, and we're going to call them the acyl. Okay, 2 carbon acyl CoA. All right, they still have this carbonyl there, right? But you have these high energy units that are going to be then used as feedstock. So basically, that's what you need to know then. And water is going to be then introduced. So this is again part of the hydrolysis kind of approach in trying to degrade these fatty molecules. So I have a little bit of obviously animation here, but the point is that the oxidation is going to remove the hydrogens, okay, from the alpha and the beta carbons. If you look up there in the cartoon, double bonds are going to form. You guys know that double bonds are very reactive, all right? And the double bond, when that forms, well, FAD accepts those electrons, right? Forms the FADH, or protons, as it were. So that's where that goes. So that's where all that is being conserved, right? Water comes in, hydration then basically renders that double bond there between the alpha and the beta carbons to be then saturated. But because it is now functionalized, now we have a hydroxyl group on one side, then that can give rise to some interesting chemistry where NAD plus starts to make its appearance, okay, in the oxidized form, and then actually get reduced in its reduction to forming the NADH. Then what that's going to do is going to steal that hydrogen from that hydroxyl group that's on the beta carbon, and then all of a sudden you have a carbonyl that's produced because there is electron rearrangement, and now you have that carbonyl, and now you have that reactive center where alpha and beta carbons can start to split, okay, especially in the presence of coenzyme A and the low energy form of it, actually, because it has that thiol group, okay? So that is just repeated. Okay, so that's basically the overall schematic and the mechanism of how that's done, but that's repeated until the fatty acid is no more. All right. So why are fatty acyl coenzyme A is created? Because that form emulates the acetyl coenzyme A produced from pyruvate. Okay, so there's a lot of similarity here. It can't be exactly because you started with a fat instead of sugar. Again, this is the acetyl coenzyme A, and we're showing the acetyl group here in purple with the rest of the coenzyme A shown in yellow. All right, you can see how there's going to be really good, similarities here among, between those. So the fatty acyl, right, coenzyme A, with however number of carbon atoms that there are in that fat molecule, is going to combine with, low energy NAD plus, low energy FAD, water, because water is going to have to be present in order to help hydrolyze and break up some of those fatty acids. It has to, in order to form that hydroxyl, which the FAD takes, and then of course you have the coenzyme A. So you have to have all of this present. When you're trying to determine, well, how many are created in terms of how many, like, what is the energy, like what is the ATP generated from a fatty acid molecule? Well, you can use this kind of molecule, molecular formula here. So just consider what you have in terms of the acetyl CoA, the NADH, and the FADH2. Do not refer to the fatty acyl CoA as a number of carbons. Really, that's just there for bookkeeping purposes, just to keep track that you have this starting molecule and that it's now going to be broken down into these other units which have currency, because right now the fatty acyl CoA does not have the currency that these others do, okay? So I think that this is important, and you divide by 2 because each of these carbon units are comprised of 2 units. That's why you divide by 2, right? You started with the chain, you want a 2 carbon unit because that basically emulates the acetyl part in the purple there up by the CoA. Okay, so that's why it's 2 carbon. And if you have 14 chain carbon divided by 2, you can get 7 units of those. So that's the whole point of this. And so this would be an example of that. You show the 1, 2, 3, 4, 5, 6, for instance, in this 14 carbon fatty acid, this myristic acid, and you know that there's going to be 6 cycles. Better have your COA present, right? Within that mitochondria. So here on the left, that's really just bookkeeping. You shouldn't even worry about that plus sign. It's just there to help you understand basically what you have. You could actually have an arrow instead of a plus sign even. But you only use relevant metabolic molecules to determine the ATP. When I say that again, that's what I mean. Only take the acetyl-CoA, the NADH, and the FADH2, because that is what we have currency for in terms of ATP units. You know that I'll use these accepted conversions, and you'll be able to identify what you got. So look, did I even touch anything with the H+? No, I didn't, because it doesn't have ATP units. Did I use the fatty acyl CoA? No, because really that's just what all this got broken down into. So really, and again, like I said, you don't even have to use a plus sign here. You could just use a yield sign. Really, that would probably be more appropriate at this part. But you have your total, which is about 94 ATPs. Yes, fatty acids have a lot of energy, and the key is being able to tap into it. So one of the issues is that a large amount of fatty acid oxidation, unfortunately, can lead to excess buildup in the liver. Okay, I don't want to say if it's too bad or not, but this is what's going to give rise to, for instance, ketosis, and this is what can be measured with ketosis strips, in the urine or even blood, and you can actually notice that in somebody's breath because these are going to form ketones, and ketones are pretty volatile. Like, for instance, acetone is volatile, and even this beta hydroxybutyrate, these are what we would consider to be ketone bodies. Even this acetoacetate has an odor to it, okay? But the point is, if you have a large amount of oxidation, you're getting a lot of these acetyl coenzymes A build up because that's how the fatty acids are being broken down. The condensation of 2 of them is going to form 4 carbon and 3 carbon ketones, all right? And these are all examples of this. Acetone is the most volatile of all these. This is what you respire. You can see that how you actually also have CO2, and if there is going to be, respiration, CO2 is going to be exhausted from the body, as well as any kind of acetone. These other acetoacetate and beta hydroxybutyrate, if they are not as volatile, and sometimes they're not going to be, they're going to be left in solution, then it's going to be circulating in the blood. This is one of the ways of detecting whether you have these molecules present in the urine or in the blood. You would say then you're in a state of ketosis, okay? Again, starvation, glucose deficiency, low carb diet, high fat diet, a keto diet would all fall under this, okay? So this is just what it is. Fatty acids aren't necessarily made to be the default currency of energy production in our bodies. We know that, right? It's sugar, carbohydrate based, but that is an example of it. The buildup of too much of that in the liver, if it doesn't have any place to go, that could maybe down the road give rise to some kind of issue with your liver, even though it does regenerate, or other organs. But I don't think the data on that is up to date on that in terms of some of these long-term effects. And in fact, some doctors, swear by this, right? The whole Dr. Atkins diet is based on this. So that could be a thing. But, overall, you're going to have less energy. And if you have less energy and you're depleting your body of those important carbs, basically, those nutrients, the glucose nutrients, which are meant to create all these other substrates that your body can use, that your mitochondria can use for a variety of other purposes, then you're probably not going to be at your best shape. So that's just something to say there. And then finally, we're talking about the catabolism of proteins. Well, guess what? It can happen, all right? All of the different amino acids can actually, be converted to these what are considered alpha keto acids, like these ketoglutarate, like pyruvate, things like this. Anything that has basically a carbonyl group, a CO double bond, right? Those are going to be considered an alpha keto acid, and it's through this transamination, meaning that the alpha amino group is removed. So in this kind of family, the C3 family, with respect to the amino acids and stuff, you're talking about alanine, cysteine, glycine, serine, threonine. They all can get converted to pyruvate. You know, some of these other ones can get converted to the different types of ketone bodies, like the acetoacetate, for instance. Of course, you know, just know that if some of these amino acids are being converted through a transamination, well, that's going to have to be eliminated. So you're going to be eliminating a lot of that more so in your urine and whatnot. So there's going to be probably an increased odor of ammonia in your urine whenever you're on a diet of high protein, or if you're in a case of starvation and your body starts to consume proteins, not only the fats, but also your own body tissue. So that's an interesting, you know, obviously, you know, scenario that you want to try to avoid. But the alpha keto acids are then converted to intermediates, and, you know, depending on what those intermediates are, then they'll just be introduced into the citric acid cycle wherever they are. I mean, if you're starting with glucose, then you're going to go through the whole citric acid cycle. But if you're starting with a different intermediate, then you're going to be admitted wherever the citric acid cycle is going to be willing to accept that intermediate metabolite. So that will just be through that way. But ATP can be produced through this way, but it's just not very efficient, okay? And really, you know, energy by proteins here, I just have this note, about 10% of caloric or energy needs are really met just by living off a diet of high protein. So it's really not good for you to be doing that. Anyway, I just wanted to end with this. This is it. That's the end of chapter 12 and the rest of the semester. And I just want to tell you, man, thanks for sticking with me, and congratulations to you, and thank you for a great semester. And I leave you with this woodcut image from my mining Bible, De Re Metallica from Agricola. This is one of the last images, and it shows that Argonauts are there, and they're sailing away. Their work is now done, and they have done whatever they needed to do. They're washing themselves in the spring and stuff, fresh water, and they're going to be on their way. And I wish you all the best as you get on your way in all of your endeavors. And so with that, I thank you and good luck.